Allison, I'd like to ask you a few questions, if that's all right. So
Yeah, for sure.
I'm not a beekeeper, but I do work on phenotypic plasticity and caste formation, and I've done a lot of genomics work. And so I was just sort of looking over your paper, your BMC genomics paper. Really great figures, by the way. Easy paper to look over. The
Thank you.
question I was really wondering, and I know that for clarity, leaving some of that out, were you surprised by any of the proteins that you found in your survey, or the genes that you found, or were some absent that you expected to be there? Are there some lines of kind of candidate gene markers that you're kind of also exploring that you didn't tell us about?
No, there aren't any ones that I'm exploring that I haven't talked about. In terms of what they actually were, the heat stress markers were totally expected. Those ones I was not surprised about at all. The only 5 proteins that were elevated in those comparisons were all called heat shock proteins. So those were totally unsurprising. For the cold shock and the pesticide exposure, I'm now honestly, I am struggling to remember exactly what those proteins were. I think one of them for the pesticides was catalase, I believe.
Right, and so I work on pesticide resistance, and so my expectation for detoxification enzymes would be that you'd see the upregulation pretty quickly, and then also sort of to some of the other speakers' points, because of, and you already know this, you talked to it at length about the destructive sampling, is that you can't tell over time what is like immediately induced versus what's induced later versus, except for, you know, taking queens at different times post-stress.
Mm-hmm. Yeah, and that's something that I will be doing, is just to get a better idea of their stability over time. I have right now, like what I haven't done that I would really like to do is like a bigger experiment, I guess, where you have like one experiment and then take queens at different times. But what I have right now, which will help a little bit, is queens that have been in a colony for a week and a half after the stress. So it's a bit extended compared to the just 2 days that I was looking at before. But ideally, I'd like a longer term experiment as well.
I think it's really ambitious to try to answer some of those bigger questions. I mean, I really appreciate, just like you said, that there's a lot of questions that are raised, and the kind of experiment that you can do is basically a snapshot in time, and that it really does generate some answers, but a lot of questions. One of the other things that I was kind of interested in is, can you impose sort of a quantitative genetic framework? Because is it possible to get queens that are sisters, or is that part of your experimental design to impose a quantitative genetics framework on the design, so that you can maybe use not just the genomics, but also sort of the relatedness to reduce the any confoundedness by genetic relatedness?
Yeah, so that's something that I would like to test like formally. Right now, in the experiment that I did this past summer, I used queens that were sisters. But what on one hand, that means that your experiment will be more sensitive because there's less noise between your experimental groups due to genetic variation. But on the other hand, what you really want is a marker that is universal, from queens from, who cares what their genetic source is. Like, what you want is a marker that will work for anybody.
Right.
So,
but in order to identify the markers, sometimes you have to take that more sort of developmental cell biology approach of, we have to eliminate variation and then go back to the variation.
Yeah, exactly. So that's why this experiment I did in the summer was that, like, very narrow, control the genetics as much as you can. And then what I'd really, really like to do is take queens from say some from Australia, some from New Zealand, some from Hawaii, some from California, and do these stress tests and see if the same markers show up. I so far I've tested, let's see, I think I tested both Californian and Hawaiian queens. I have to go back and check my notes, but I had two different stocks for the heat stress markers, and those two were elevated in both of those different stocks, we'll call them. So that was promising, at least. But yeah, it really needs to be done across like a wider range.
Ali, I have a question for you. Are you on those queens? Are you running PCR on any of them to look at virus is in those queens and see if that also affects any of these markers?
Yeah, particularly after Michelle published that paper about the heat shock response being antiviral.
Yes, exactly.
Yeah, all of a sudden that became very pertinent. So actually, yeah, I think we have a lot of samples waiting to be run at the National Bee Diagnostic Center.
That'd be great. I mean, nobody really has a good, I mean, Michelle's done work, seeing how the colonies as a whole, get a virus, coming in and going out all the time that change. And I don't know whether individual queens, get sick, a little bit sick from time to time and then get better and then get sick again. So that would certainly be of interest to see. And that may be one of the reasons we see these colonies recovering. When the queen looks bad and then she recovers, was she just sick for a little while?
Yeah, maybe. I think there's something a lot more complicated going on.
Could well be.
But yeah, I'm struggling to remember exactly how many we have, but there's something like we have 50 or 60 queens that are going to be tested for viruses and nosema too.
Mm-hmm.
And those are the same queens that I have the proteomic data for. So I'll be able to see if those markers covary with the viruses or not.
So, hey, are you gonna, with nosema testing, are you gonna look for nosema species?
Yeah, I suppose we will, because we started trying to do it ourselves with the microscope, and we're just having an impossible time. There were so many spore-like objects. We're like, is that a spore or is that not a spore? So we decided to go molecular with it.
Okay.
So presumably the primers they distinguish between the different species.
Okay, the reason is that me looking at queens and looking at spores, and once you've looked at enough spores, the species are pretty easy to tell apart. We've gone from when Nosema apis was considered to be a major problem with queens, and nowadays we don't see much Nosema ceranae in the queens, at least at the spore visual count. So that'd be interesting information to see whether Nosema, now that we should to largely Nosema ceranae, is still as much of an issue to queens.
Mm-hmm.
You guys,
yeah, we will be able to answer that.
There's some questions in the chat. Can you guys, in the Q&A, can you guys see that? Or I can read them too if that's easier.
Yeah, I'm opening it up right now.
And I think this is for anybody who's still around, if Steve or Endany or Kevin, Allison or Randy, you guys feel free to answer these. The first one I see is there's two from Cheryl Burrows and one from Jenna Gibbons and one from William Holmes.
Allie, you want to do it or you want me to do that one?
The one from
about the electric fences?
Oh, yeah, I would just be guessing. I would say there's probably not a negative effect on the
Well, there's definitely a safe distance to have the fence from the hive, and that is when you bend over, you don't shock your butt.
Yeah. So, I think they're asking about the bees, though.
Yes, I know. We have fences on most all of our colonies, our yards, and we don't see any adverse effect from them.
This one about what would we study if we weren't limited by funding, that's kind of interesting. Gosh, I'd have to think about it, I think.
Yeah, it's an interesting question because there are applied researchers who are more applied, and then others that are just more, biological, want to learn more about the bees. So for me, it's definitely the applied research. Number one, how to control varroa, and either by selective breeding or treatments, or management techniques. That would be the thing I'd really be interested in. Actually, what I am doing, but I would expand that.
Certainly, one of the things that keeps coming up in Washington when we were doing the pollination task force this year is, number one, varroa mite control, and then number two, all of these diseases that you guys have. And I think, there's a lot of research going on that, but from the beekeeper's perspective, I guess they feel like there's not a lot of solutions that they have in their hands right now. I don't know. That's what it seems
Right.
like.
That's true. I have a list actually I made up for grad students of 2 pages of unanswered screaming questions that we don't know about honeybees, honeybee biology, and I have it broken down into all different kinds of categories that it's freely disseminated. I think I actually have it on my website, but anybody's looking for a list of unanswered questions, I've got one.
I don't know if there's a shortage of those.
No.
I think that for me, if I wasn't limited by funding, I'm really, really interested in this crazy biological system that the honeybee queens have where they can keep sperm alive for so freaking long.
Oh, yeah.
And, it's rivaled only by ants. The ants actually surpass the queens. They can keep sperm alive for decades. So I think that there could be some really cool opportunities there for studying sperm health and sperm function more generally. And what I would really like to do is develop some kind of like spermatheca in a tube system where that would allow me to test like what happens to the sperm when I add say different hormones or different proteins, or even like subtract some proteins. There are ways that I think it would be actually feasible to do that. And in that way you could really like drill down into exactly what effect do all these different things have on the sperm's longevity over time, and like come up with a way to rival, I guess, what the queen is able to do in terms of keeping them alive for years and years. So that really interests me because a lot of the same things that affect honeybee sperm viability also affect mammalian sperm viability. And I think that coming up with some kind of in vitro, like sperm in a tube system to do these really specific experiments might actually could, theoretically help better understand some aspects of human health too. So
Are you doing much brainstorming with Boris Bear about that?
Yeah. I've been in contact with Boris right from the beginning.
Okay, yeah. I mean, yes, he was studying this back when I visited him in 2008, and he was just all over it then. So yeah, I'm glad to see you guys talking. Mm-hmm. There's one here from, tell me about the process of registration for the oxalic acid and glycerin. I am very deep into conversation with USDA right now, and I'm submitting them a list of questions that I've told them I'm going to be publishing in American Bee Journal of what the hurdles. There's a whole bunch of stuff that I have by phone and email of the hurdles that they're facing and the pushback they're getting from various agencies. So, but I think beekeepers need to have USDA themselves answer this. So I've told them I'm going to be I'm going to be publishing his answers to this, so we can figure it out. See whether the beekeeping industry needs to petition the USDA and EPA directly of how important this is. The easiest way for the EPA to get around this is to just register the IUN cap strips, and then at that point they'll say, oh yeah, you already have this. You don't need anything else. Somebody's going to have to spend a ton of money to come up with a new one. What I'm pushing for is a generic additional application method that any beekeeper can just feel free to make this themselves, which would allow the beekeeping community to experiment with coming up with just what works well in your area. So we're deeply involved in this right now, engaged in this discussion. And screen bottom boards. That should be from some local beekeeper up there who has experience with that. When screen bottom boards first got popular, we built 500 of them in our operation, and we have phased them mostly all out now. We did not see much benefit in our operations. So that'd be for somebody else to answer.
I've used screen bottom boards forever. I never put a cover over my screen bottom board. They're open all winter. Bees do fine. I don't know if there's an advantage or a disadvantage of having a screen bottom board or not. Up here where we are on the cold side, we like lots of ventilation in the winter, because if we don't get it, the metabolism of the bees, they exhale, and that water condenses in the top of the hive and then drips down through. So if you have good ventilation with lots of airflow, you don't seem to get that type of condensation in your hive and get wet bees and freeze them out in the winter. So I think that's the main reason I do it. Some people say it's good for, because the mites drop all the way through and hit the ground and can't crawl back up in the hive. I don't know if that has any effect. I haven't seen any data on it. Maybe Randy has some, but.
Oh yeah, very easy experiment to do.
Right.
You take a dry sticky board and you take your finger and you make a ring of Vaseline around the outside perimeter. And you see how many mites get stuck in the ring of Vaseline. That tells you how many were still alive when they drop down.
And how many is that? Is that
insignificant number?
Okay. So when they drop, they're not alive anyway. So
right.
Okay.
Healthy mice don't tend to drop.
Right. So anyway, you know, a screen bottom board, I don't know. I think it's probably just personal preference. I'm not sure it has a huge effect.
That's the feedback I'm getting, is even on the moisture regulation, it's largely personal preference and because it makes sense rather than supportive data.
Right. And because all my hives have screen bottom boards.
You're the normal beekeeper. There's no control group. So
exactly.
Yes.
You know, yeah, I don't know. Yeah, like I said, it's just the way it's been done, and that's not really a good reason to do it that way.
Well spoken.
Did any of the other researchers have their thoughts on what they would research if they weren't limited? Anthony, how about you?
I don't know. I guess the thing that I've always been dismayed at is the state of bee breeding. How you can pick up a cattle catalog and you know the expected breeding values of what you have, and we still don't have like a breed. Like, it just strikes me that, but that's a hard, and I don't know if it's a research problem. I don't know exactly what the research question is. So
that's a practical problem, and that's awesome. You're absolutely right. I mean, my family was dairy farmers, and you could go to the Badger breeder in Wisconsin and they could tell you exactly what you were going to get. And you can't do that with bees. And in fact, I find it amazing that in the US, like around us, you know, people are saying, oh, I have Caucasians, or I have Carniolans, or I have Italians. They're American bees. We haven't imported bees.
Well, but the good news, and I saw that Steve sort of came on, is that you do have, you know, the best program at WSU at Kraken.
Right. You know, and well, you know, what our little local club has been trying to do is actually breed a local bee, you know, so we're trying not to bring in as many packages. So people are trying to get genetics that are more, you know, more suitable for our area. I don't know how well that's working, but because we still bring in an awful lot of packages, which brings in, you know, a lot of diversity from outside. So
yeah, Kevin, actually I don't know if the Inland Empire beekeepers are still doing it, but they were one of the groups that really took to heart this idea of sort of trying to breed their own bees and use, you know, stock that we had provided over the years. So I don't know if they're still doing that or not.
yeah, I haven't heard anything from them about that lately. I know they were doing it a few years ago, but you know, we still see all the local groups that sell packages. You know, they're bringing in, you know, they probably bring in 1,000+ packages every year, and 1,000 packages kind of swamps out any breeding attempt that we do. So, you know, we've talked about, you know, in our club saying, hey, you know, we'd really like people not, you know, let's just let everything die off that's not going to make it, and then let's propagate from what we have. But that's
Yeah, the problem you have, Kevin, you know, as we all know up here, is we can't make queens very early. So
right.
and people want their, you know, want to start their colonies in the spring, and
Right.
Yeah.
But in our club, you know, our club apiary last year, we made, you know, and we didn't even make a huge attempt. I think we made 11 or 12 additional hives that we sold off. You can propagate bees fairly quickly, you know, and you know, so you think about, you know, you start with 3 or 4, you can get 12. The next year you're up to 30 or 40. You know, you can propagate fairly quickly.
Right.
But that doesn't support the 1,000 that people want every year. But that also tells me that 1,000 hives are dying every year because they're bringing them in every year.
This is typical. And in answer to Adoni's question, it's the mating. So if you're not going to do instrumental insemination on all those queens, you need to have isolated mating yards, or as I have is, and USDA data support this. They've done 2 studies that if you absolutely are flooding your area where you're mating with your own stock, you can control 95% of the matings. One of the things that I saw years ago, I was driving up to Idaho and going through Nevada. You have desert for miles and miles. Then you have these little green valleys that are oases where they have an alfalfa farm or 2, and then desert for miles. Each one of those is an isolated mating island. So throughout the United States, there's these isolated areas. We do a pumpkin pollination out in the middle of the Nevada desert with nothing, no honeybees for miles and miles. And then there's a place where they have surface water close to the surface, cheap, and there's a big sod farm and farm there. And we're the only honeybees going there at all. Same thing in our upper elevation yard. So it's not impossible to find isolated mating islands where you can totally control the genetics. It would just take it. But the thing is, no one's demanding it. And business responds to demand. And right now, as you say, the beekeepers are not demanding from the queen producers to take the extra step, whereas the dairy farmers reward them for taking the extra step. Beekeepers do not reward them. Beekeepers will buy any queen available in the spring because they're desperate for it. So there's no incentive for the breeders to go the extra step and seriously get engaged in these breeding programs. And that's one of the reasons I'm trying to walk the walk, and just to see how difficult it is to actually selectively breed for mite resistance. And if I can be successful at it, then I'll be able to lay out how others can do it, which that's my only interest in it.